Hi everybody, this is Dr. A. In our third and last microbiology basics video, we are going to look at how do we identify these little microbes. So first, we do microscopic observation. So bacteria are incredibly small, colorless, and invisible to the naked eye. So we have to use microscopes and use stain to make them visible so that we can identify them. So stains are used to make the bacteria stand out from the background and enable microbiologists to study their morphological feature and distinguish between different groups of bacteria. So morphology is shape. So for example, if you looked at this slide, you would see not only are there plenty of white cells around, but these bacteria are stained dark purple, which is going to be characterized later as you'll see as gram-positive. You can see that their shape is long, elongated, like rod-like, and you can see that they're in chains. And so those are already some morphological features that you can identify from the bacteria of this sample. So the gram stain is the most frequently used stain in bacteriology. So it is used to identify bacteria, okay? And almost, exclusively bacteria. It will stain fungi and stuff, but we use it to identify bacteria. We cannot use a gram stain to identify viruses. So the results can be obtained in a matter of minutes. It does not take very long to do a gram stain. Once the specimen is fixed to a slide, the staining procedures literally only takes 2 to 3 minutes. And can differentiate bacteria into one of two groups. There are either gram-positive or gram-negative. If they stain this deep purple color, they're considered to be gram-positive. If they stain the pink counterstain color, they are gram-negative. That provides a preliminary classification of those bacteria into two different groups. And then you combine that with their morphology, which it can be characterized, for example, as rods or cocci if they're round, and then are they in clusters, are they in chains, etc. That can already give the physician a clue as to what it is. So for example, if you saw, like is seen in this slide, the purple ones here, so they would be gram-positive, and they are cocci because they're a little round, and they're in clusters. So you see gram-positive cocci in clusters, you would start assuming and thinking along the line of this is a type of staph. So it could be a staph infection. And so anyway, and if it was a gram-positive cocci in chains, then you may think strep. And so there are ways we can kind of get a presumptive identification, or at least a path to run on for the physician to treat that infection. So the diagnosis of many infectious disease is still based on the isolation and identification of the pathogenic microorganisms on culture medium. And so that is the traditional way. It's the way we've been doing it for many years. We have learned to automate this process, and we have added some more molecular techniques to it and some newer identification techniques, but this is still kind of like the basics, and we're going to go over this. So the culture is the growth of microorganisms on artificial media, such as these agar plates shown here. So we want these microorganisms to grow to where we can see the colonies with the naked eye, and then we can do tests on the colonies. The culture medium is a mixture of nutrients on which the microorganisms are grown. I'm going to go over those here in a minute. But culture media can be solid, semi-solid, or liquid. So these agar plates are going to be considered solid. Semi-solid would be more like the characteristic of gel, if you will. And then liquid would be liquid, like a broth. The solid media are best used to observe the colony size, shape, and color, because it's solid, it's flat, so the bacteria grow on top of it, and the way they grow it can be quite distinctive. Some of them will make really tight, round, neat colonies, and they'll be a specific color. Some of them that are motile may just like swarm and spread, so have colonies that are more flat and spread out. And so that can also help identify the type of bacteria that's growing. The semi-solid or liquid media are used to observe things like gas production, odor, or changes in pH if there's a color indicator in the liquid. So what is agar? Agar is a seaweed extract that is used extensively in the preparation of solid media. The advantage is it's liquid when heated and it is solid when cooled. So you can prepare it with proper nutrients and get it to boiling temperature and it'll be sterile, and then you can pour it in a plate and it will take the shape of the plate the petri dish that it's poured into. Put the lid on the plate, let it cool. Once it reaches room temperature, then it's solid. It does not affect bacterial growth, meaning that the bacteria grow on top of the agar, but it doesn't break the agar down. Okay. It's an excellent base for the nutrient media. So the nutrient media are the nutrients that we give the bacteria to grow. And with agar, you can make plates, as shown here. Slants right here. This is a slant, and stabs. So with a stab, you would actually stab the culture into the gel here, into the agar. And all of those are done with agar as the base. Then we can classify media into different types. So supportive media contains nutrients that will allow most non-fastidious organisms to grow at a normal rate. So what is non-fastidious? It means just not picky, if you will. Fastidious organisms are just picky eaters that have to have conditions just right for them to grow with the right type of nutrients, etc. So these guys that can grow on supportive media usually are just run-of-the-mill. They grow very happy to grow on agar, and they grow pretty quickly and easily. Enriched media contains additional nutrients to help the fastidious organisms to grow. So it provides the proper nutrition for the picky eaters in the bacterial world. And then media can also be selective and or differential. So selective media, it selectively inhibits the growth of some type of organisms while promoting the growth of another. And it uses dyes, antibiotics, or other chemical compounds to do that. And so it selects for a category of bacteria or for a certain type of bacteria. And then differential media, they contain dyes or indicators that will give colonies of certain organisms a distinctive look or an easily recognizable characteristic. So it would be bright pink, or it will be black, or it will be dark purple, or it's something that stands out, that when you look at the plate, you can say, oh, I know what that is because of the color, the change. And then the other organisms that don't do that reaction, maybe they're just pale in color or just white or something. So you can really see the difference between the two organisms and easily identify them. So some of the commonly used media, we're going to start with sheep blood agar. It is a supportive agar right here. Lots of stuff can grow on it. Actually, most of your run-of-the-mill pathogens that infect the body can grow on sheep blood agar. Chocolate agar, though, is enriched, so the fastidious ones can grow on chocolate agar. It's actually related to blood agar. It's just that the blood cells are lysed in the chocolate agar, and so the nutrients are more easily accessible. It's more enriched. Then you have Microscopic agar. It is both selective and differential. So it is selective, as in it will only allow gram-negative bacteria to grow on it, and it's differential because certain ones like lactose fermenters like E. coli will turn a bright pink on that Microscopic agar, whereas the other gram-negative bacteria that are non-lactose fermenters will stay kind of just a pale color. Eosin methylene blue, or EMB agar, is also selective and differential. That one is also selective for gram-negative bacteria, meaning it inhibits the gram-positive from growing. And then, for example, here, E. coli also has a distinctive light green sheen. But other bacteria that are that ferment sucrose and mannose and other things can, maybe it's fructose also. Anyway, some of the fermenters can turn a dark purple, almost black color. Then you have Thermartin agar. So that one is enriched and selective. So it actually looks like chocolate agar. It's enriched, but it's also selective in that the only type of organisms that can grow on Thermartin agar are the Neisseria. especially Neisseria gonorrhea and Neisseria meningitis. Then we have thyroglycolate broth. It is enriched. I don't have a picture of it here. Just portray a tube that looks like chicken broth that's pretty clear. That would be thyroglycolate broth. We often use it after we've swabbed inoculated the cultures on the plates. We also often inoculate a thyroglycolate broth for testing later on in case something happened with the plates or things didn't work out or we need to go back for more sample. Then we have the saburo-dextrose agar with antibiotics that is selective, so it promotes the growth of fungi while inhibiting bacterial growth. So this is saburo's agar. And then we also have the Lowenstein-Jensen media. It's this pretty teal one. It is enriched and it is used to cultivate an isolated Mycobacterium species, which is really hard to grow and very slow growing. And so that one allows it to grow. Then you have the culture requirements. So the environmental requirements also needed for successful bacterial cultivation are going to be proper temperature. So usually we set incubators at body temperature because pathogens live in the body and so they're used to body temperature and they do best at body temperature. We also give them an adequate supply of oxygen, or in the case of anaerobes, we can put them in pouches that are going to remove all the oxygen and fill it with CO2, so we get the appropriate gas environment for them. And we have to also make sure that everything's at the correct pH. That would be more a function of the media used being at the right pH. And then we can identify them. So once the specimens are plated or inoculated on several different types of growth media, so different plates, right? And depending on what type of specimen is sent, we set up different agar plates. Okay, and inoculating is so it's getting the bacteria culture from the sample, getting the bacteria on there, and then we go and put them in the incubator, and we hope that they grow. And once there's growth on the plate, then we can look and we can isolate and identify what's growing on the plate. So this one has got a bunch of different types of bacteria because you can see a difference. They look different here, and so there's a bunch of different, this is quite heavily, I would say, either cross-contaminated or it's a very big mix of bacteria. And this is more representative of what you would find from an environmental swab. But you could technically, each different type, you could do tests on each type of these colonies of bacteria and identify what each one of them is. So once that organism has been successfully cultured and you see colonies on the plate, those colonies then can be used for biochemical and antimicrobial susceptibility testing. So that the process of getting, plating it and getting growth can take up to 24, 48 hours. Some, if they're fast growing, you may see some colonies as soon as within 12 hours. If it's slow grower, it can take, a couple of days. We don't call them completely negative and no growth until we've let them grow for at least 5 days for your average cultures. And so then when you take, those cultures, those colonies that have grown on the plates, you identify, I want to identify this colony. If you have a couple of them, a couple different types, you may want to set up testing for each one. And you make a solution of it, and then you inoculate that solution in these biochemical plates. And so these biochemical tests are tests that are based on the presence and measurement of an enzyme. We can see does bacteria produce have the catalase enzyme to produce certain reactions, or the oxidase enzyme, or indole or urease tests. And so these all looking to see if the bacteria has a specific enzyme, because we know certain types of bacteria have certain enzymes. And then it's also based on the presence and measurement of different metabolic pathways or byproducts. Are they oxidating something or fermenting certain sugars? Can they degrade certain amino acids? So we basically kind of look at their eating habits and metabolism habits And based on the pattern of reaction in all these biochemical tests, what's positive, what's negative, the computer looks at all of it and matches it up, and it says, okay, based on the pattern here, then this is identified as this specific type of microbe. And so this used to be done by heavily manual methods. A lot of this has been automated, so that, again, once you've inoculated that, it takes, again, the bacteria have to grow and then ferment or, metabolize different things. So it takes a few hours, up to half a day for this to happen. But you let them do its thing, and then you look at the machine will look at the reactions, read the reactions, and then spit out an identification for that bacteria. Then these biochemical test results, when you combine them with the microscopic, the gram stain, what it looked like on the culture plates, then put it all together, you can verify that it's a biochemical ID was given, does fit everything else, and you can usually report it out. There are some newer techniques, such as the multi-TOF, that can allow the use for mass spectrometry to identify microbes that have been cultured without the use of biochemical testing. So you have to go through the first process of planing it on media and getting the colonies, but then you can take the colonies and get direct identification using this multi-TOF mass spectrometry technique. There are also PCR type identification panels that use the microbe's genetic information to identify the infection, and those are usually run on the initial specimen. So they're run on a sputum or on a fecal test. A fecal sample, and it tests for a whole panel of microbes, including usually viruses, bacteria, and fungi that are likely to be causing infection, and it tests for all of it all at once. And then lastly, usually the biochemical testing is coupled with an antimicrobial susceptibility test. So when we set up the biochemical test, we also set up this. And so this is testing performed to determine whether or not that particular organism is susceptible to various antimicrobial agents. So they are reported as that bacteria is either susceptible, resistant, or intermediate for each of the antibiotics. And so it will say, penicillin may be resistant, but methicillin sensitive or something like that. And there's a whole long list of antibiotics, and it will tell you for each one of them for that particular bacteria if it's susceptible, resistant, or intermediate. So if it's susceptible, then it indicates that the patient will most likely respond to treatment with that specific antimicrobial agent, and that will clear the infection. If it is resistant, it indicates that the treatment with that antimicrobial agent will not work. It's going to fail. That bacteria is resistant to it. That antimicrobial agents are not going to do anything to it. And the intermediate, it's kind of in between the two. So maybe a high dose would work, or maybe you find or use an antimicrobial where you have a susceptible result. And so this can also help identify microorganisms such as MRSA, so that's methicillin-resistant Staph aureus. You can identify it with the biochemical testing of Staph aureus, but the antimicrobial susceptibility test then allows you to tell you that it's not just regular Staph, it's actually MRSA. So that can also key into de-identification. And then it is the job of pharmacy and the nursing staff together, once they get these antimicrobial susceptibility tests on those culture results, to make sure that the patient's antimicrobial therapy that was started is indeed appropriate according to the results. So if you put them on an antibiotic and find out that the microbe's resistant to it, then you need to switch the patient to a different antibiotic. But if they put the patient on an antibiotic and find out the patient is susceptible, or the microbe is susceptible to that antibiotic, then the patient can stay on that antibiotic, and you should already be seeing improvement. And this is because if you look at the whole testing process, so from getting the sample to plating it to letting it grow to seeing the colonies and then doing the biochemical test and the antimicrobial susceptibility test, it can take 48 hours from specimen collection to the time you have results. Well, if there's an active infection, you don't want to wait 48 hours to treat it. So the physician is going to treat the infection based on some of the presumptive information, as like signs and symptoms and where the infection is located, and then maybe some the gram stain results, which can be done on the front end and resulted out really quickly. And so they'll start them on something, but you really don't know for sure until you get that ID and susceptibility test as to whether or not that treatment has been actually appropriate for that patient. And so there you go. That wraps up the whole identification and susceptibility testing for microbiology basics, and I thank you for your attention.